Welcome everyone to the International Myeloma Foundation's webinar, Top Myeloma Research, presented at ASCO, EHA, and IMWG 2025, The Patient Perspective. I'm Robin Toohey, Vice President Patient Support at the IMF. And if you're not familiar with these acronyms, ASCO is the American Society of Clinical Oncology, EHA is the European Hematology Association, and then IMWG is the IMF's International Myeloma Working Group. And speaking of the IMF, as a reminder, we are the global leader in multiple myeloma, and our mission is improving the quality of life of myeloma patients while working towards prevention and a cure. We also have a beautiful vision, and that is to realize a world where every myeloma patient can live life to the fullest, unburdened by the disease. The IMF was founded 35 years ago now, and our 4 pillars still stand strong: research, support, education, and advocacy. So tonight, we are going to hear from Dr. Joe Mikhael, as well as 3 patient advocates that I'm honored to introduce you to tonight. Todd Kennedy was diagnosed with myeloma in 2017. Todd is an active patient advocate and support group leader. He also strives to ensure that myeloma research is patient-centered by serving on advisory boards for clinical trials and research collaboratives affiliated with the National Cancer Institute and American Society of Hematology. Todd recently attended the ASCO annual meeting. Next up on our panel is Barbara Leonardi. Barbara was diagnosed with myeloma in 2019. Her first line of treatment was a clinical trial. In 2020, she became actively involved in patient advocacy through the Polish organization Foundation Carita and the European umbrella group Myeloma Patients Europe. Barbara's main focus areas are education and communication, and currently serves as president of the board at Myeloma Patients Europe. Next in the picture is Dr. Joseph Mikhael. He is the IMF's chief medical officer who will be guiding the patients through the conversation. And last up in this photo is Mikhael Toohey. Mikhael was diagnosed with myeloma in 2000. That quick math makes him a 25-year myeloma survivor. He has been on a variety of treatments, including a clinical trial. Mikhael is a longtime volunteer patient advocate, and in 2001, he co-founded and continues to co-lead a myeloma support group in Prospect, Connecticut. Mikhael recently attended the International Myeloma Working Group meetings. Before we get started, it's always wonderful for us to thank our sponsors this evening is AbbVie, Johnson & Johnson, Regeneron, and Sanofi. And to all of you out there that want to engage with this panel and Dr. Joe, please look here on the Q&A in the window. You can ask your questions and to the host and the panelists. Now, please remember, if we don't answer all of your questions today, we are expecting a very robust, engaged evening tonight. Please remember, you can always reach out to our info line, and that number is 1-800-452-CURE 2873. And we definitely want to hear from you. Your feedback on our survey is imperative to us continuously improving our programs. So at the end of the meeting, please stay on. A pop-up will come onto your computer, and that will be a very short survey. It will only take a moment to complete for you. So now it's time to get started. And one more little reminder. So after tonight's program, as all our webinars, we have it recorded and available on the International Myeloma Foundation website. That's myeloma.org. And if you click on that top panel that says publications and videos, and you click on videos, that's where you'll find tonight's recording, as well as so many others. So now it's actually my pleasure to turn it over to Chief Medical Officer Dr. Joseph Mikhael.
Thank you so much, Robin. I just appreciate so much being here. It's always a pleasure. I have the privilege of giving so many talks a year, but the ones like this with patients, for patients, is fantastic. And I just want to also give a special shout out to Todd, to Barbara, and to Michael for joining us tonight. For those who may not know, it's not just a coincidence that Robin and Michael have the same last name. Michael is indeed Robin's husband, and Robin, you are a beautiful example to us, not only in what you do at the IMF, but how you support Michael through his journey. And it's just absolutely wonderful. You are such a beautiful example of what a care partner can and should be. So thank you everyone for joining me tonight. As we say, we are locked and loaded. Like, this is going to be a jam-packed next hour. So I hope you've got your espresso or your water or whatever it is you like to consume in the evening. It may be an adult beverage, but let's dive in. This really starts by me explaining that this is a tale of three meetings, as was mentioned, which is why we're gathered today to go over what has come out of myeloma research at these three great meetings. We had the ASCO meeting as described, where we had 45,000 people in person. We had the International Myeloma Working Group. Now, there are about 350 members of the IMWG, but a little over 100 of us gather for this summit right before the European meeting. And so 200 in total with our guests and so on. We met in Milan, in hot, steamy Milan, I might add. And then we had the European Hematology Congress in Milan, Italy, which itself was a bit of a hot yoga studio. And there we had about 18,000 individuals. So at these venues, we have tremendous research that's presented, and we're going to focus on 5 areas today. And the approach that we're going to take, and I see questions already streaming in, which makes me very happy, is that I'm going to give a quick introduction to each of these topics based on the data that was presented, and then I'm going to turn to Michael, to Barbara, to Todd in various orders, and just get their impression and their take on it. Because as we always say, it's one thing to hear Dr. Joe's version of it, it's another thing to hear it from the patient. For those of you who were with us a couple of weeks ago, we did a similar program with a physician alongside of me, Dr. Sagar Lonial, and indeed Dr. Beth Feynman, an advanced nurse practitioner. And we had a conversation about these same topics at a certain level. This one, we've really tried to focus in such a way that no matter where you are as a patient in the journey, that you'll be able to understand the implications of these research. So we're going to talk about smoldering myeloma, frontline therapy, or newly diagnosed multiple myeloma, patients who have early relapse, which we define as, generally speaking, patients who've had 1 to 3 prior lines of therapy. And a line of therapy is basically treatment that someone's given until the disease grows again. Late relapse, which we typically define as at least 4 prior lines of therapy. And then we have a little bonus topic, which is an important one because it was a big part of our discussion at the International Myeloma Working Group Summit, which is we've just literally hot off the press. I got to tell you, in myeloma, we published some huge papers in the last 4 weeks. And this is one of them where we now have a new definition of high-risk multiple myeloma. So let's talk about smoldering myeloma. Now, as most of you know, we have a spectrum where patients either have MGUS or monoclonal gammopathy of undetermined significance. I saw a patient just an hour ago in my clinic who has had now MGUS for 15 years that we've been following him. And that just means, of course, that someone has a little bit of that abnormal protein, but not enough that we would want to treat it. And their risk of them becoming active myeloma is about 1% per year. At the other end of the spectrum, of course, we have myeloma, which we all know about and unfortunately needs to be treated, although you're going to hear about the great treatments we have for it. Well, in between, we have smoldering myeloma. And even within smoldering, there's a huge spectrum. There are people on one end of smoldering that really probably don't need treatment, and there are people at the other end of smoldering that are really about to become active myeloma. And this has been a hot area of study because we want to catch cancer as early as possible. And there are different tools and models. And one of the things that came out very clearly, and Michael, I may get you to comment on it earlier because you heard some of this discussion at the Working Group Summit, we all realize that myeloma is not really one disease, and it can be very different, and it's hard to measure it. Some of you have heard me say, I call myeloma like a crime scene. There's not one piece of evidence that tells you the whole story. There's lots of pieces of evidence, but we're trying to collect the evidence to know, does someone have high-risk smoldering myeloma or standard risk smoldering or low-risk smoldering myeloma? Meaning, do they have a likelihood that within the next 2 years they're going to get active myeloma? If I told you you're going to have active myeloma in the next year or two, chances are you might want treatment. Other people might say, well, what's that risk, doc? Is it only 20%? Or maybe I'll wait. Well, but if it's 80%, you might want to go ahead. And one of the models we've developed is this what's called 20-20 model that we'll make mention of later, that when someone has smoldering myeloma, which we define as more than 10%, but if they have 20% plasma cells in their bone marrow, if they have an M spike over 2, and if their free light chain ratio of the so-called involved over uninvolved light chains is over 20, when we have those three factors, if you have 2 or 3 of them, you were considered to have high-risk smoldering myeloma. And this was really important because of the Aquila trial that we got a bit of an update on at ASCO and EHA this year, that looked at taking those people with high-risk smoldering and saying, okay, right now we typically actively monitor those patients. Let's do that, but let's do it carefully. Blood work every 3 months, PET scan every year, bone marrows every 2 years, so that we're really watching as close as a hawk as we should. And those are the people in the red line here. And at the 5-year mark, you can see the number of patients who unfortunately have already progressed to multiple myeloma. So down to 40.8% of patients still did not progress to myeloma, which tells you that over half of them did. Meanwhile, the other option was, let's give them daratumumab or Darzalex, the single agent, and let's give it for 3 years and see what happens. And in that group, at the 5-year mark, 63% of them still hadn't developed myeloma. Now, that means that 37% of them had developed myeloma, but we can see a big difference between the two curves. If we specifically look at those patients that have the 2220, as I just showed you a minute ago, you can again see the big difference that on average, those people who are actively monitored, they developed multiple myeloma within 2 years, but the daratumumab arm hadn't quite reached that yet. So what's my take on this? Well, this was a massive study that really is changing the way we're thinking about multiple myeloma. It doesn't mean that every single patient with smoldering myeloma, you must take this drug, but it is now an option. Instead of saying, let's wait and watch, as it were, we can say, what if we give you this milder intervention that probably won't significantly impact your quality of life? We may be able to kick the can down the road. And, I've had some patients who've talked to me even about it this week, who are in a situation where they say, doc, if I can kick this myeloma down the road 5, 6, 7 years, this may be great. I may or maybe never have to face multiple myeloma. On the other hand, some patients might say, no, I might wait and watch. And then if myeloma looks like it's really coming, then we'll give full treatment. And we're studying this more. We're now looking at other clinical trials with an even more intense look at this. But I'm going to stop talking now, and I'm going to turn to my friends here on the panel and say, Todd, as a patient, you have active multiple myeloma, as we heard. We know you've been treated already, but what's your take on this? Is Dr. Joe blowing smoke here, or what? I mean, is this, we make a conclusion that the most important thing to do is have a discussion with the patient. I mean, how does that resonate with you?
Well, first of all, thanks for having me. It's really an honor to be with you, and to be in the room when this data was presented at ASCO was so inspiring and educational. But I think for the smoldering patient, and those that I interact with, many don't know whether they are high-risk smoldering or whether they are standard-risk smoldering. So I think that conversation you just had about the 2220 is really very important. And if people don't know, I would suggest that they probably need to have that conversation with your doctor to help inform that decision. And I think that the Aquila data is very compelling. It could delay progression, but it is a personal decision. like you said, and there's no judgment in that decision. But one thing that inspires me is the idea, like you said, of kicking the can down the road. Because as we get further into this conversation and talk about the pace of innovation in frontline therapy, if you were able to delay progression for 3, 4, who knows how long, the conversation we'll be having in a meeting like this in years from now will be very different. We'll have new options, new insights and things. So that idea of kicking the can down the road is very attractive to me. But again, it's a very personal decision for, I think, each and every patient. But understand the risk, I think, is important.
Oh, thanks, Todd. That's actually really helpful. In fact, I had a patient tell me the same thing today, where they often say, well, doc, if my disease comes back in 3 years from now, what will you treat me with? And I'm like, well, I can't tell you, That's like saying, what's your favorite, what are you going to have at your favorite restaurant in 6 months from now? Well, it depends what's on the menu, right? And the menu continues to change. Barbara, if I can ask you for a moment here quickly, you and the work at Myeloma Patients Europe, I mean, you've done some extraordinary work in surveying patients. You have such an engaged network, and you've even published some fantastic data around this. What is your take on this and the importance of having the conversation with the patient about whether or not they want to be treated or want to continue to be actively monitored?
Yeah, I think it's always good to have options. And as you know, every patient is different. So for some, the approach of watch and wait is perfect. They are happy with that. While for the others, it's super stressful. So they would prefer to start some treatment. And that's why I think it's fantastic to see that we have an option now. And also it has been, it received a positive opinion from EMA last week. So hopefully we can see it in our European countries very soon. So I really think it's important that people have a choice, and then, of course, it leads to the shared decision making. So to have this conversation, to have a dialogue, what works for me, right? What is better for my situation, my context, my family life, etc. Also the mental state. But what I also would like to add here is the importance of the early diagnosis. Because we can still see that there is a problem here, that not many GPs, not many family doctors are aware, and patients are still diagnosed too late. So they are not smoldering anymore, they are active. So unfortunately, they miss this opportunity.
I think you make an excellent point. I mean, it's been a huge part of our work. And as you know, in the work we've done, particularly with our Empower program, to educate the primary care world and to educate the lay public about what the signs and symptoms are of myeloma to catch it earlier. I was very happy to see that positive comment from the EMA. And we're also, as you know, here in the US, waiting for an FDA approval, but we had a very positive discussion at what's called the ODAC, or the Oncology Drug Advisory Committee, where this was discussed. Well, before we close this topic, Michael, let me turn it to you, because I know you're such an active follower of so many things in myeloma, and thankfully it's been years since you were in the smoldering phase, because we heard you're a 25-year survivor, which every time I say it, I love saying it because we're so grateful for you. But what's your take on this?
Oh, sorry, Dr. Joe, we just lost power. There's a raging storm coming through here. I didn't even hear the question you just asked me. My take on it, I thought Todd and Barbara covered it perfectly. The one thing I would add is, you're talking about kicking the can down the road. And a treatment like DARA, which to me seems like a pretty decent therapy to take with limited side effects. So to be able to, move it down the road further before, and if you're not that close, like you and Dr. Loniel had the discussion last week about, if someone's that close to being full-blown myeloma, then he would treat them with 4 drugs. So it's a, incredibly individual, whoever's personal decision, how about you go. But I like the DARA study. I think to take a treatment that has very limited side effects is, a good way to go.
Yeah, I mean, I think that was a critical piece of the Aquila trial. You said that so nicely, Michael, that it was that sweet spot of enough and not too much, right? Because there are different strategies. And to close off this session, I'll remind people we are studying. I already see a question from my friend Steve Weinstein asking, are there other trials? We are now looking at doublet trials and triplet trials. Believe it or not, there's a clinical trial looking at CAR T-cell therapy in high-risk smoldering myeloma with the concept of saying, can we extinguish the disease even even, and maybe even more than kick it down the road, maybe kick it off the road, right? so that people never have it again. And this is a really intense area of work. And Barbara said it so nicely. We want to catch cancer earlier as much as possible. All right, well, we need to keep rolling here because, time goes quick when we're having these exciting discussions. There was maybe the most talked about abstract in the frontline setting. I got to argue that later there's the most talked about abstract overall, but maybe the most talked about abstract in the frontline setting was this MIDAS study. And I know there's a lot on the slide, but follow me on this, because if you capture this slide, you understand the concept we're thinking about. Todd mentioned that we've had huge progress in frontline therapy. One of the biggest steps we've seen is now we go typically from a triplet to a quadruplet therapy in myeloma, whether someone's going to transplant or not. And that we really established last year in the transplant ineligible population and eligible population. This French study, the MIDAS study, takes it a step further and says, you know what? What if we treat someone with a quadruplet, in this case, isatuximab, carfilzomib is the K, Revlimid is the R, or Kyprolis is the K, Revlimid is the R, and dexamethasone, what they think to perhaps be the most potent quad we have, and treat someone for 6 cycles, then decide on what to do based on their MRD status. I know it says standard and high risk, which can be a bit confusing, but just think of it as being either MRD negative above or MRD positive below. And saying, okay, if you are MRD negative, let's then test the hypothesis. Do you need a transplant or not? So they either gave them more ISA-KRD or gave them more ISA-KRD with a transplant. And what was fascinating is there wasn't a big difference between those two arms. Now, as I'll say in a moment, that doesn't necessarily mean we don't do transplant anymore. If someone's MRD negative, I had this exact conversation with a patient today who's scheduling his transplant. But it is giving us a hint that maybe there might be a population we may not have to. At the bottom part of this of the study, they took those patients who did not achieve MRD negativity, so were MRD positive, and they need to perhaps be treated a bit more intensely. So they randomized them to either, kind of like above, giving them a transplant with some ISA-KRD or giving them 2 transplants. And again, interestingly, in this group, in the second bullet that you see here, the 2 transplants didn't seem to get people into any deeper response. Now, MRD negativity, we love. We know that MRD, or minimal residual disease, is kind of, the test that looks for tiny amounts of myeloma left in the bone marrow. If you can believe it, we can look for 1 in a million or 1 in 10 million cells to see if there's a myeloma cell. But this study, we think, is so important because it's helping us see that maybe that tool of MRD might help us determine who needs a transplant and who may not need a transplant. Now, I don't want to prejudge it because I think we all need longer-term follow-up because things could change later. But this may be a potential with time to reduce the need of transplant. As I joked with someone the other day, I guess this means I shouldn't be investing in transplant stock, but I don't want to give up on something that has helped so many patients for so many years. The other study that I wanted to mention was an update to the Perseus trial. The Perseus trial was the study that led to us using 4 drugs or quadruplets in frontline therapy. And again, I don't want to go too much detail here, but basically compared patients at the top getting 3 drugs, VRD, Velcade, Revlimid, dexamethasone, or adding D to it, the daratumumab, before the transplant and then after the transplant. If you look at the maintenance phase, this is the part that we are getting an update on. That they had the strategy of saying, instead of just giving Revlimid maintenance, which is typically what we do in most patients, maybe not in high-risk patients, but let's keep it to standard risk patients for now, they add daratumumab to it for up to 2 years. I won't go into all the details of the study, but the whole idea is saying, let's give you 2 drugs instead of 1, but at the 2-year mark, we can stop the daratumumab and just keep the Revlimid. And what they showed was, if you just look on the right-hand side here, those people that had that 4 drugs up front and a transplant, 2 drug maintenance, 55% of them not only got into MRD negativity, but stayed MRD negative for 2 years. So Ken, to answer your question, I'm not talking about smoldering myeloma. This is active myeloma now. We're talking about patients that have true myeloma. So you have over half of people MRD negative for 2 years. We've never seen that number before. The latest estimate is that if Perseus, as we're going to continue to follow it on a mathematical model, this might mean that the average time that someone will have myeloma before it progresses is actually that may live with myeloma is going to be over 14 years. Lastly, there was the GMMG concept trial. And I highlighted this one because Todd is a bit of an expert in helping us understand high risk versus standard risk myeloma. And we're going to come to this at the very end, but I'll remind you that about 20 to 25% of patients have so-called high-risk myeloma. It just means their myeloma has a bit more of horsepower and it can grow back quickly. And we've struggled sometimes with how to treat high-risk myeloma because the disease comes back. And the German group here said, tell you what we do. Let's take high-risk patients. Let's give them 4 drugs with ESA-KRD. Let's give them a transplant. Then let's give them more ESA-KRD, and eventually we drop the DEX, which is, I'm always happy to see, down with DEX, but then they continue that ASA-KR for a long period of time. And of course, it's not concluded yet, but I know it's hard sometimes looking at these curves, but what this curve on the right shows us, sadly, every time the curve drops, it means a patient has passed away. But if you look out at how that curve is shaped and you look at the bottom line there, it says 6-year overall survival rate of 69%. Historically, patients with high-risk myeloma didn't live more than 5 years. Now we have over 2/3 of patients living more than 6 years. So I think it's teaching us that we have to be more aggressive in patients with high-risk myeloma. And this may be the key here. They showed that the patients in blue were MRD negative. The patients in red were not MRD negative. That if MRD negative is a powerful tool, it is incredibly powerful when it comes to high-risk patients, meaning that we want to get our high-risk patients into MRD negativity, because if we don't, it's very likely their disease is going to come back quickly. So it's important to use that as a target for us. So what's Dr. Joe's take on this? Well, quadruplets have really become the standard of care now. And if you're here tonight and you're being treated, this is important to know and important to have a conversation with your doctor, or if you're newly diagnosed and soon to be treated, that really, I just met with a patient about 2 hours ago who was just diagnosed in the last few weeks, and he's starting his DVRD next week. And he said, my doctor said he watched a video of you talking about 4 drugs versus 3 drugs. You know, this is so important for us to understand. I'm not saying that MRD is ready for prime time, that we shouldn't yet be making major decisions in frontline therapy based on MRD, but these studies are pointing us in that direction. And maybe someday there will be some patients that we can avoid transplant because of it. In particular, for patients with high-risk myeloma, we really should be looking at that as a goal. And then a couple of last comments that I didn't have time to show you the studies with, but it was fascinating to me how many of these studies are using carfilzomib or Kyprolis in the frontline setting. It's not something we've done a lot of in the past, but we may be doing more of that in the future. And I didn't show it, but there is even a clinical trial of bringing the drug that we hope to have back with us as a relapse drug very soon, belantamab. In some countries, it's already been approved again, was even being used in the frontline setting, and bispecific antibodies, and even someday CAR T-cell therapy. But that's going to be for our next year's session. So when the 4 of us are back here again, or the 5 of us are back here again next year, we can talk more about that. But let me turn to the crowd and think. And Barbara, maybe I'll start with you. I know this is like a flood of information, but what do you see in all of these studies around frontline therapy?
Yes, it is a lot of information, but what is really striking for me is that finally we are seeing the end of the one-size-fits-all approach. So now we are seeing more subgroups, more personalized treatment, and I think MRD, which is not a standard of care yet, but I think it will be, because we can see that it is working, that the patients, because what we want a patient to be treated, but not over-treated or under-treated. And I think these studies show that MRD can help the clinician to decide, okay, this patient, it's enough for him or her, right? We don't need to burden them with more drugs that they need. Or, as you said, Dr. Joe, does this person need a stem cell transplant, which is also quite a hard procedure for the body. So I think the MRD.
So beautifully said. And I love the way you talked about options, because really right now, and that's one of the reasons why I mentioned even the carfilzomib, obviously in different countries and jurisdictions are the coverage and so on. But in general, I mean, we have 2 monoclonal antibodies, we have 2 proteasome inhibitors, as well as the immunomodulatory drug and dexamethasone, meaning we have both Darzalex and Sarclisa. We have both carfilzomib and bortezomib, or Kyprolis and Velcade. And so we do have options for our patients. Mikhael, what's your take on this? This is a little different than what you were treated with 25 years ago, brother.
Oh, absolutely. High dose dex, got ready for my transplant. Not fun. The quad is incredibly impressive. I mean, 2 years ago when we were at ASH, I was seeking out, why are we going to 4 drugs versus 3? And every myeloma expert I found at ASH that year, I approached them and asked them, including you, and everyone said 4 drugs. Being you could take the DARA afterwards until you're MRD negative, then pull the DARA back so you're not refractory to it. I think it's a great plan, and the numbers speak for themselves. I mean, it's really impressive.
And I think what it does too, Mikhael, is it to me, in my mind, I often describe it this way. I say that, we know that the biology of myeloma is complicated. I sometimes say it's like weeds in the lawn. Even though at 112 degrees today, I'm not sure my lawn is very happy here in Phoenix. But if you have different weeds, you need a mix of weed killer to get rid of them all. You can't just use one, and that multiple mechanisms of action has really proven itself in the frontline setting. And I know we don't have time to get into a lot of detail, but we're also learning that sometimes even using lower doses of multiple classes of drugs is more effective than trying to use full doses of fewer classes. Todd, what's your take on this?
Yeah, I think the first point on the slide is so important. Quads are the standard of care, clearly. And it says transplant eligible, but I know there was also an update on Cepheus and things too. So I think it's also standard of care in transplant ineligible, for most patients, not just high risk, not just transplant eligible, but for a pretty broad population. And the other part that I think some patients miss, we talk so much about quad therapy, but when you looked at those trend lines on Perseus and other quad therapy trials, it's not just quad therapy up front. It's also, like you said, dual maintenance, or in Midas, it's triplet for a long time after. So it's not just quad therapy for 4 or 6 cycles. That dual maintenance part is probably very important as well. And I think people need to remember that that's how you got the kind of extraordinary response that you said with, like in Perseus, maybe 14 years, 17 years, crazy remission out of that type of regimen. And then that also then speaks to me, like you have a recipe per se, like with Perseus, that could give you more than a decade, perhaps, of remission. And that includes transplant. So I agree. I'm not ready to write transplant off right now, because there's a proven formula that, again, then could kick the can down the road to give you future options. But at the same time, I love Midas. I thought that was the most fascinating design that I've seen in a trial in a long time. And I love the 6 weeks, the 6 cycles up front with a harvest at cycle 3 too. So
yeah, we want to make sure
exciting stuff.
Yeah, we have the ability to collect the stem cells. I mean, I think one way to summarize as we move into our next, is I also don't want to intimidate people to say that it's always more. Paradoxically, when we use more drugs up front, we can then de-escalate them down. And it may mean really in the long run that patients are getting less treatment, because as they're staying just on lenalidomide alone or Revlimid alone, and sometimes we get them even off of that. My goal would be have a strategy that ultimately we can have people off treatment. I always say it, my favorite drug is nothing, nada. I love giving people nothing. And when we extinguish as much of the disease as we can with these kinds of combinations, we have a better chance of getting down to nothing. All right, we've only covered 2 topics. We've got 3 more, so let's turn into early relapse. And I wanted to include this study because it was fascinating. It was using the drug we know, isatuximab or Sarclisa, but now giving it in a different way. And I've got a little picture of it here, and I've had a chance to interact with this thing. It's a little almost, this was presented by our French colleagues, and Xavier Leloup said it was like a little snail, like a little escargot. that gets attached to the skin, and it just a little button is pushed. There's no, external needle involved. It doesn't actually have to formally be administered by a nurse. And what happens is when you push that button, there's a tiny little 30 gauge needle, which is really quite small, and it then goes into the skin. And over about 12 to 15 minutes, it administers the drug. And this is fascinating, and I don't want to comment too much because I got 3 patients surrounding me here, so I want their input on it in a few minutes from now. But the whole idea here is that this may be a much smoother and easier way to deliver isatuximab, which we now give IV. Thankfully, we do give Darzalex or daratumumab subcutaneously, but this may be even opening the door for people to self-administer this at home, because it doesn't require necessarily a nursing assessment and a nursing expertise to deliver it. That being said, of course, here in the US, it's going to be an uphill battle for that. But I think about how this potentially could affect other people in the world. And when they studied this, they did survey the patients. Not only did the drug work just as well subcutaneous in this now what we call on-body injector as it did intravenously, but patients were more satisfied with it, and we really didn't have, thankfully, any unusual safety signals. The other studies that we can't help but mention are the belantamab studies, because Bela is going to hopefully come back very soon. So belantamab is an antibody drug conjugate. What do I mean by that? It's a drug that hooks on to the myeloma cell through a target, the myeloma cell called BCMA, and then it's got a drug conjugate, which means it's got this backpack of a toxin that it can drop into the cell to help destroy the cell. This drug was approved a number of years ago. It got removed from the market and is now, we believe, coming back based on these 2 studies. This first one is DREAM7, where interestingly, you saw earlier a lot of studies that were 4 versus 3 drugs. This was 3 versus 3 in the relapse setting, which is tough to do because typically we do 3 versus 2. But this drug went head-to-head against daratumumab by giving it belantamab or belamaf, as it's written here, with bortezomib index versus daratumumab bortezomib index. And we saw that those patients actually had a remarkable 3-year on average remission. This is in relapse myeloma. This is not frontline myeloma. So to get 3 years out of something, as you see here, 35.6 months, was really remarkable. What was updated at our recent meeting was this was also very evident in patients with 1 or more high-risk cytogenetic abnormalities. Those features that we talked about earlier that bring us to high risk, we saw that benefit. So my take on this is a couple of things. One, the on-body injector, I think is fascinating to me, and I'm going to be interested to see what the 3 of you think about in a moment. What do you think of the idea of sticking on this little device onto your body for about 15 minutes as you get the drug? I really believe that belantamab will be reapproved soon, and we anticipate and hope that will happen. There is an ODAC coming up in the very near future, just in a couple of weeks. And I think we're going to have the option of combining it with bortezomib, as I showed you. There was another study where it was combined also with pomalidomide. For time's sake, I didn't show that to you. But this is a very active drug. And what's fascinating about it, although there are some side effects associated with it and some eye-related symptoms that we have to work through, we're getting better at managing those. Often we give this drug every 8 or 12 weeks. I have a patient of mine who literally, she gets this drug every 4 months. And so to have that kind of infrequent treatment that allows people to maintain their quality of life, I think is going to be important. And there may be patients in particular who will not be eligible for CAR T that this would be great for. And then lastly, I didn't have time to go in a lot of discussion, but we saw some we saw some abstracts with probably the newest and soon to be approved bispecific antibody, like literally any minute. I check my phone several times a day because we may hear about this literally any minute. Linvoseltamab is going to be another new bispecific antibody that's soon to be approved. And we saw it being tested in early relapse in different combinations. But let me turn to the group here. Michael, let me start with you. What do you think about this on-body injector device? I know that you've had a chance to read a little bit about it and see it in pictures. What do you think, my friend?
I actually was impressed. A 30 gauge needle, compared to someone that's going into the doctor's office getting pricked and needles and everything. That's, we were talking about this last week, or at GMAN, the Australian clinical trial was doing daratumumab at home, and we were talking, saying that most likely wouldn't happen in the United States, and it probably is an uphill battle here with the OBI, but it really could be done at home. I would think quite easily if it was willing to take the chance to, like you don't need a nurse to really, like a sub-Q shot. So I think it's really interesting. It has a pretty good future.
Yeah, it's interesting you commented on the 30 gauge needle. People may not be familiar with it. I'm sure there's some people Googling that right now to actually get the actual size. But this is a tiny, tiny needle. This is like, the lead pencil. It's tiny, tiny. Barbara, what is your thought around this? I know that in parts of Europe in particular, there has been greater opportunity to deliver certain drugs at home. And it's not just about delivering at home, of course. It's really about shortening the IV time and shortening the time that someone might have in the clinic. But what has been your take on the on-body injector device?
Yes, as you mentioned, Myeloma Patients Europe did some research around the quality of life, and also in particular on one group of patients, the working population. So for them, such a device would be a fantastic solution because, it can allow you to run your life in the standard way, which means going to work, taking care of your kids or parents, or whatever your family situation is. Plus, you don't need to go to a hospital to spend there one day, right? So we really think it would dramatically improve the quality of life.
Wonderful. Todd, maybe I can ask you about the belantamab question because I know you're so well informed and have been keeping up to it. And I see a couple of good questions that have come in that I get to answer after you have a chance to speak. But what do you think about these Dream 7 and Dream 8 studies and the potential of bringing belantamab back?
I am like you. I am watching my phone for the good news, hopefully, when we get that decision. And I think it is probably inevitable, and it's very needed, especially, like you said, in that second point around, it'll be something that's important especially for frail patients, maybe not eligible for CAR T. And I love the fact that it partners well with other agents. And I think that was one of the other things that showed up in, the Linvo trial, which will also be another option, just thinking about all these different combinations. Typically, we get these agents as single agents, but a lot of the research coming out of ASCO and EHA was how do these things work well with others? And so I think Bella is going to be an amazing contribution.
Yeah, I think you captured that well. I love how Barbara used that phrase earlier. It's not one size fits all.
Right.
As I say it all the time, I don't treat myeloma, I treat people, right? And every person has to be looked at uniquely and differently. And I really do think it's so many times the nurse practitioner, Susie, who I have the privilege of working with, often says, oh, this would be a perfect patient for belantamab. They may live 2 or 3 hours away and the frequency of visits and so on will make it easier for them. So I really do think there will be a lot of patients who would benefit. Robin, with your permission, I'll answer a couple of quick questions here. Why was belantamab removed? What was the problem? Well, typically in the approval process here, Catherine, great question. A drug gets approved typically when it's given as monotherapy, as Todd had mentioned, in a smaller study, while we wait for a larger study to be done later. And the first larger study done with belantamab, it didn't show that the drug was worse than the comparator, but it wasn't significantly better. And so that's usually the bar that has to be reached for a drug to be approved. So we're withdrawn. But we knew at the time that these 2 trials, Dream 7 and Dream 8, that I briefly mentioned, were in the works. And now these have come out so strongly positive, we anticipate it'll return. And in answer to Mindy's great question, yes, it can be problematic for the eyes. We did learn when we give the drug every 3 to 4 weeks, over half of patients will have some eye-related symptoms, namely blurriness of vision and a change in their visual acuity. We are coming to appreciate, like with almost every drug in myeloma, as we use it, we have to change the dosing strategy. In fact, I often say there's literally no drug we use now the way it was first introduced, and we're using the drug less frequently, every 8 or 12 weeks, and we're seeing considerably less of those eye-related side effects. And then lastly, for our anonymous attendee, just to make it clear, I mentioned it earlier, but when I say the phrase early relapse, we're referring to patients that have only 1 to 3 prior lines of therapy. So typically, there's still a number of drugs or classes of drugs they've not yet seen.
Hey, Dr. Joe, can I add just one more point on that?
Yeah, please, Todd, go ahead.
I think it was really important, and I think our listeners probably would appreciate that slide you mentioned around the patients that had high-risk features and actually did really well on Bela. And I know that there was CARTITUDE-4 that was there too and showed that, CARVYKTI did well in that patient population that was high risk as well. So I think we're going to talk about high risk, and people maybe find out that they're high risk, and sometimes they would maybe then feel like all hope is lost. But these studies are showing even if you have some of those high risk features, you can have very, very good, strong response.
Yeah, I think that's very important. And it's part of our evolution of thinking, right? Is that we want to reduce the fraction of patients that really have true high risk. And, Larry, you make a great comment here and comment/question in the Q&A about, it used to be that we didn't want to use our whole arsenal at once, and we like to save things for later. I often say saving the best for last is marvelous for a romantic movie, but not for myeloma. You are absolutely correct. We are learning that what we do early on in frontline therapy at first line and as second line therapy, sometimes I call it the 1-2 punch. When you look at the two things that someone is treated with at their frontline therapy and at their first relapse, that may be more prognostic than any risk score you have. And so we don't want to save the best for last when myeloma becomes more aggressive, when myeloma has had more time, if you will, to be resistant to the treatments we have. We want to use our best therapies earlier on as much as possible. I wish we had more time to talk about all the other things that are happening in early relapse, like using more CAR T-cell therapy, like using bispecific antibodies, although right now they're only approved in late relapse. But Robin will pull me off the stage if I don't keep moving. So let's move on to section 4, which is late relapse. Now, if you have been napping so far, please wake up for this, because I'm going to argue that this was the most talked about abstract at ASCO and EHA. When we have all these abstracts presented, they're usually presented in the myeloma session. But for each of these meetings, because these are cancer meetings or hematologic meetings, we often have what's called a plenary session, which means these are the top abstracts across all diseases in hematology. This was one of the plenary from the European Hematology Association from EHA. And it was the long-term follow-up of CARTITUDE-1. And I'll remind people that CARTITUDE-1 is where we gave cilta-cel. The name here in full is ciltacabtagene autoleucel, but we call it cilta-cel, or the trade name is CARVYKTI. And CARVYKTI was given in these patients, and I'll remind you, these patients had, and this is why we call it late relapse, on average 6 prior lines of therapy. So these are people that basically were running out of options. This is a group of patients who I would have predicted, to be very fair at the time, would have been expected to live approximately a year longer because we have so few options for them. But the drum roll, please, the key slide that was presented was this slide. And I know this curve doesn't look that compelling, but let me tell you what it means. What it means is that 1/3 of patients, 33% of patients, after getting a single infusion of CARVYKTI, 5 years later, are still disease-free. Now, think about that for a moment. Here are people that are expected, sadly, to live a year, and 1/3 of them are 5 years without disease and without needing further treatment. This is caused us to even think about, how do we define cure? I'm not saying these patients are cured, but we have to think about how cure might ultimately happen. And if a patient is off all treatment and without disease, that's at least steps towards that cure. I define cure, I'll define it a little bit later, a bit more simply, but really this was, this even landed on the front page of the New York Times, or in the New York Times, because this was such a hot topic when Pete Voorhees presented it. And then I secondly want to highlight what was also maybe the second most talked about abstract, which was a tri-specific antibody. And I know this can be a little bit confusing, but let's remember what are bispecific antibodies. Bispecific antibodies are drugs that have 2 arms. That's why they're called bi. One arm grabs onto the myeloma. One arm grabs onto a patient's T cell, one of their soldier cells in the body, and engages it to destroy the myeloma. This is now a I don't have 3 arms, but this is a tri-specific where now it hooks on to 2 parts of myeloma, 2 targets on myeloma, and then the third arm engages the T cell. And the concept and the idea is saying, can we grab onto the myeloma more tightly or more effectively, more precisely, so that we hit the myeloma and not the other good cells in the body? And it was fascinating as this was presented that we saw patients, and again, it's a little bit early on to make huge conclusions, but here we have patients with 100% response rate. I mean, just almost hard to believe at what's called the recommended phase 2 dose of the RP2D. So we always, with using a new drug, we build up the dose to make sure we're not going too high to begin with. And so at this recommended dose, we saw every patient respond to this tri-specific. And again, without going into too much detail, what really I found interesting was that this drug hooks onto 2 parts of the myeloma cell, what's called BCMA, which we talked about before, B cell maturation antigen, and then another thing called GPRC5D. We do have a bispecific antibody that targets GPRC5D. It's called talquetamab or Talvey. We know that sometimes people can have some tough side effects with this. It can affect their taste, it can affect their appetite, their skin, their nails. But when the drug was given this way, we saw actually less side effects than when we would typically give the bispecific of Talvey. And we think that that had to do partly with the fact that this drug is now given once every 4 weeks instead of once every 2 weeks. So can we hit that sweet spot where we give enough drug, more, if you will, targeted or precise, but not so much that it causes the side effects? And so time is going to tell. We need more time. There was another tri-specific, for example, that targeted CD38 and BCMA also that I was fascinated by. But I wanted us to introduce this principle of saying maybe a tri-specific antibody could be better than using subsequent bispecific antibodies. On the subject of using 2 bispecific antibodies, we had what's called a late-breaking abstract at EHA, the European meeting, that Shaji Kumar presented. Many of you would know Shaji from Mayo Clinic. He's on our scientific advisory board. And here what they did is it wasn't a tri-specific. It is actually the 2 drugs that they're giving at the same time. But what they wanted to do was to use it in arguably the hardest place to treat myeloma, which is when patients have a very aggressive form of myeloma called extramedullary disease. The medulla is an old word we use for the inside of our bones, our bone marrow, and that's typically where myeloma lives. But in some patients, myeloma lives outside the bone marrow. We call that extramedullary. For myeloma to live outside the bone marrow, to live outside of its usual environment, it typically means that it's pretty aggressive. And so we thought, okay, let's take these patients with very aggressive myeloma and let's give them these 2 drugs together and see what happens. And interestingly, we saw almost 80% response rate in this very aggressive disease. When we compare it to when we gave those drugs individually, the response rate was in the 40% range. So we almost doubled it by giving the 2 drugs together. This is not a new thing for myeloma. I remember the days, I'm old enough to have been around back, and of course, Michael, you would remember this because you were treated with high-dose dex by itself. But remember when we had both Velcade and then Revlimid came out, people are saying, oh, wow, what is it going to be? Are we going to start with Velcade or are we start with Revlimid? And we learned that if you give Velcade and then you give Revlimid, you get a certain amount of response from each of them. But in fact, when you combine them together, you get even more. There's that sort of concept of synergy. And here, I think we see that same principle in the redirect trial. All right, the last major trial I'm going to talk about today is the Imagine-1 study. There was a lot of excitement around this because it's a new CAR T-cell therapy. And this was an interesting study to us for 2 reasons. One, we now see a response rate of well over 90%. Which was very, very exciting. We want to be able to treat patients. This CAR T is targeted a little bit differently than the current CAR Ts we have. It is BCMA directed, but it's a little bit different. But the second thing that was important about this study, not only did we see a good depth of response and duration here, meaning at 1 year, we still had 80% of people in remission, but we also saw less of the neurological side effects that we sometimes see with CAR T-cell therapy, and specifically with Carvykti or Cilta-cel. Now, I don't want to, decide who's going to win the Super Bowl after halftime. Although I guess last year, I'm sure you could have figured out who's going to win the Super Bowl at halftime. I'm sorry, Michael, that it wasn't your Vikings. But anyway, but we don't want to predict things too early. But it is really encouraging to see that we may not have to experience some of these neurological side effects, especially the ones that are more severe, that are more delayed, things like Parkinson's and others that we see, albeit in a small fraction of patients, less than 1%. It's very important for that 1% of patients. So, wow, a lot to unpack from late relapse. But in summary, the CARTITUDE-1 study showed us really remarkable results with a third of patients disease-free at 5 years, and then starting us to think about how we define cure. We have seen these tri-specifics, in particular the one that targets BCMA and GPRC5D, to be quite impressive in its response rate and less side effects. I mentioned the other tri-specific, I think, is also going to be a particularly important target for us to use. Using TEC and TAL together is really impressive in extramedullary disease. And I'm excited about multiple future drugs coming, including this anito-cel, the CAR T that I just showed you. But wow, there's a lot there. I don't know what you think about this, Barbara, but a lot to take in here in late relapse. What do you think?
Very briefly, every myeloma patient has two dreams, CAR T and a cure. And I hope we are getting there. When I was starting my myeloma journey over 5 years ago, CAR T was like science fiction. And now it is happening. So that's it.
It is. I mean, honestly, when I first described to people, I'm like, okay, we're going to take these T cells out of you. We're going to train them to know what your myeloma looks like and multiply them in lab and give them back to you. They're like, Dr. Joe, are you describing like a Star Trek movie or something? Or are you actually talking about what we do? I mean, it is really remarkable what we're doing. Michael, what is your take on these late relapse studies?
I think it's amazing. The anito-cel, the way it's constructed with the D-domain binder, seems to be the reason you're not getting a neurotoxicity. That the teclistamab, talquetamab, that's impressive with extramedullary, but how about the tri-specific in that case? Because you're hitting the same targets, correct?
Yeah, no, I mean, I think that's something we're going to have to look at with time. And I'm sure with time, like we did with the combination of TEC and TAL, we had multiple, what we call subgroups, that with time, they'll look at a subgroup. I really like the way you've mentioned that, because I do think that will be particularly important in that population, because it can be very challenging at times to treat patients with that big burden of disease. And that's kind of an interesting thing about these drugs, is that they have that ability to reach those more difficult to hit areas. Todd, what are your thoughts here?
Well, I was in the room when they presented that CARTITUDE-1 stuff in Chicago with you, and I had to restrain myself. You know, everybody tries to keep it all low and mellow, but I wanted to stand up and cheer. You know, and because in that concluding slide, he talked about, Dr. Voorhees presented, you know, it represents the potential curative power of this CAR T. And potential is a good word. You know, we can't declare yet, but that New York Times article that came out as soon as I came back from Chicago, my next door neighbor showed me his New York Times and he says, "Are you cured?" I said, "No, I'm not." But I love the fact that it's part of the conversation. And I think that'll just inspire hope, and that'll push people to get more educated and connected and all the like. And then the tri-specific was just extraordinary as well. To think you could get maybe similar results with good tolerability and with once monthly dosing, that is an extraordinary advancement, I think, in late relapse.
Yeah, I make sure I want to emphasize that, because I mean, if I think of like my nirvana for treating patients, right, and the three of you are patients, you know better than all of us, is can I give something less frequently? Can I give something with less side effects? And can I give something that works better? I mean, can you have your cake and eat it too? And I guess I think we're baking some pretty good cakes here, and I hope that we'll be able to to be able to do even better. All right, I'm getting hungry, so we got to get towards the end here. We know we're scheduled for an hour, but we're going to go a little bit over because we have one more section to cover, and then these questions have been streaming in, so we'll jump into some of those questions. I will remind everybody that we do try to answer as many questions as possible. I actually spent the whole day in the studio yesterday in Los Angeles, and a lot of the time I spent was answering a lot of these questions that come in. So always send us questions at the IMF. We're happy to help you through the info line, through the chat bot. We have our own Milo AI chat bot that you can reach, and then of course I'll do my best to answer them. This really leaves me with one slide, and I was a little nervous at first to put this slide up because people look at this and go, Dr. Joe, can you please speak to me in English and not in science? And for those of you who aren't as familiar with this, please don't be intimidated by this slide. But for some of you nerds out there, myself included, my wife always tells me that she loves that I'm a nerd, and so my nerd gene comes out here. You might be interested in this. Let me just quickly put this in context. We've said from the start of this session today that myeloma has a spectrum, as it were, meaning even there's from MGUS to smoldering to active myeloma. But even if you just look at patients who have true active myeloma, there's a huge spectrum. Thank God Michael is with us. It almost makes me emotional, Michael, but thank God Michael's with us 25 years after his diagnosis. That's wonderful. I wish that were true of all of our myeloma patients. We still lose myeloma patients. I lost a myeloma patient a year ago who died within 6 months of her diagnosis. And it still stings my soul every day when I think about her and how her family has lost this incredible individual. And so within that spectrum, we want to give the best treatment for everybody. Barbara said it beautifully earlier. We don't want to overtreat and we don't want to undertreat. How do I match the right treatment to the patient? And that's what we're learning. And we think MRD is going to help us with that. One of the things that helps us is if we can define as best we can, we know it's not perfect, but as best we can, who has the most aggressive myeloma, who may need the most aggressive treatment. Those are people, it's going to be harder for me to give them nada because they may need more treatment. Now, granted, with CAR T, we may be able to stop treatment in some patients, but who can we define as high risk? And I know this is a gamish of all sorts of letters and numbers for some of you, and I'm not going to go through detail, but what we did is we came together as the International Myeloma Working Group and the International Myeloma Society and said, let's work together to define high-risk myeloma, and we came up with these 5 things. If you have one of these 5 buckets, then you would be defined as high risk. And I say, I'm not going to go through them all. Some of them you may have heard about, things like deletion 17p. There's interesting what's called a T53 mutation. So this is something that we need advanced testing to do. Regular FISH studies don't pick it up. And there's a lot to do with hair changes in chromosome 1. But the idea here is not so much that you as a patient need to memorize this, as much as you talk to your doctor about having high risk, of understanding what that may or may not mean, and that your doctor does the right test to figure it out, because we are now starting to do clinical trials. I already showed you one in the GMMG concept study that specifically targets patients with a high-risk disease. So my take on this high-risk disease definition is that, as I've mentioned, it's very challenging in myeloma when we have people that just seem to have this bonfire-like myeloma that we can't take care of. This is going to need new testing, as I've said. But if we can define that roughly 20% of people that have the high-risk myeloma, we may be able to ensure that we don't overtreat some and undertreat others. But we do need more options. We need to study this more, because even within that 20%, there's a group of so-called uber high risk or ultra high risk myeloma that we need to attack. Now, Todd, I know you have been a tremendous proponent of ensuring that patients know their risk status. I wonder some of your thoughts here before we dive into the Q&A.
I'm a nerd like you, Dr. Joe, but I know you don't need to be a nerd. I think the important part is, like you said, just have a conversation. I have a lot of conversations with other myeloma patients, and some don't know whether they're higher standard risk, and this definition is more precise. So I would, especially if you have an expert that can really dig into the complexity of these this new definition. I think that's important to just initiate the conversation, because it does factor into that shared decision making around if I'm high risk, that could influence my decision on transplant if and when, or dual maintenance, or a clinical trial. All these things we've been talking about up to this point could be impacted if you knew with precision what your risk status was. I think it's important.
Yeah, I mean, I think you captured that really well. I was sharing with a patient today, and we have a high risk myeloma support group that has been started recently. And shout out to Becky Bosley, who's helped put this together. And when I was describing it to that group, I say having high risk myeloma is like having the red sports car. It's more likely to drive quickly, right? It has that greater horsepower or potential to come back more quickly. But sometimes, I told a patient today, sometimes a Prius is passing the Porsche. It doesn't always behave that way. So these models aren't always perfect. But what we have learned, as I think we showed you a few times today, that we may often have to treat high risk a bit more aggressively. We didn't get time to show the slides, Barbara. I know it's a study that you were interested in because it was based in Poland. But one of the other studies that I didn't show us today was the Atlas study that gave two drugs in maintenance. And this time, instead of adding daratumumab or Darzalex to Revlimid, they added Kyprolis or carfilzomib to Revlimid. And we saw the benefit of adding that second drug, and the benefit is particularly seen in the high-risk population. This builds on the work that was done before in the so-called Forte study. So I really do think that understanding your risk status is important as we go forward. Now, there is so much more that we could say, but let's do this. I'm going to quickly give us some final takeaways, and then Robin, with your permission, we'll do a bit of a rapid fire on trying to answer some of these questions. And then I'd love our three patients to sort of give their final thoughts before we wrap up. But in quick summary of what we've covered today, frontline therapy has evolved. That literally all patients, transplant eligible or ineligible, are going to be treated with a quadruplet. Now, there are some who may not be able to be given the 4 drugs. I never want to use the word all, but the majority of patients, we can do that. And we're seeing much better outcomes with it. And looking ahead to the future, we do think MRD or minimal residual disease will continue to be used and may even help us determine who may or may not need a transplant. CAR T-cell therapy is arguably the most potent therapy we have in myeloma. And it's wonderful that we are seeing, at least in a very significant subset of a third of patients, they're disease-free 5 years later. They've gotten that one treatment, and 5 years later, they're still without disease. But wow, there is a lot more to come. Whether it's the little escargot that we put on our skin to deliver sarclisa, whether it's belantamab, whether it's linvoseltamab, whether it's bringing CAR T-cell therapy up front, new CAR Ts, even CAR Ts that have different targets. We're even developing something called allo CAR T, where we don't collect your own T cells. We have T cells collected from healthy donors. And when you go to Costco, you can pick. No, you can't buy them at Costco. But we're thinking that we could have literally off the shelf CAR T that we could just infuse in patients. These are the exciting times that we're seeing in multiple myeloma. But before we dive into the questions, let me just turn to each of our 3 patients, give you a chance to make kind of a final statement or final thoughts on all that we've said today. I'll start with you, Michael.
The landscape has certainly changed from when I was diagnosed. It's really amazing, all the therapies that are on their way, the trispecifics. I know we talk about the trispecifics a lot. It's just, like you said, there's not one treatment that starts out and continues that way. I mean, look at high-dose Dex. Everything has changed. And the bispecifics, when they first came out, there were some pretty scary side effects. But the more that you're working with them and the data comes out, they're becoming effective therapies with not as many side effects. So it's really amazing compared to when I was diagnosed. It was basically a transplant and high-dose Dex. And today, I mean, how many abstracts do we have at ASH last year? It's just unbelievable, all the research that has gone on in multiple myeloma, and we're very fortunate for it.
Wonderful. Beautifully said, Michael. Barbara, your final thoughts.
Well, the medical research, the science is so advanced. It's absolutely amazing. I just hope that our health systems, our authorities will be able to follow and to use it to the benefit of patients.
Wonderful. And by the way, I have to give a special shout out to Barbara. For those of you who are aware of world time, she is now past 1 a.m. her time. I don't know about you, but you look fresh. You look great to me. You look like you're ready for more. So we particularly
Because I'm enjoying this meeting so much.
we appreciate you joining us at this late hour. Todd, final thoughts from you, brother.
I would just say science is awesome. You know, it really is. And I'm so grateful to you, Dr. Joe, to all your colleagues that are driving this research forward, and to every single patient that's been in those clinical trials that we talked about today, and those that will sign up for future clinical trials so we get there faster. I think my hope meter has never been higher than it is right now. And I know it inspires me. I hope it inspires others to just maybe rewatch this, get further educated, get empowered, get an expert, and live and celebrate life. We've got a lot to look forward to.
So, I mean, I couldn't say it better, Todd. I just love the way you capture things. You're absolutely right. I hope your hope meter continues to grow as we grow forward. Well, Robin, I'm going to turn it back to you in about 2 minutes. Let me rapid fire try and answer some of these great questions that have come in. I encourage people to keep sending them in. We keep record of all these questions, and don't forget to interact with all of our social media outlets where you'll see me answer many of these questions, whether it's on Instagram, whether it's on X, whether it's on LinkedIn, and we'll be hosting Facebook Lives to answer questions where we get to say, ask Dr. Joe anything. But a few questions that came in. Thoughts on CAR T for newly diagnosed? Yes, there are great studies. The CARTITUDE-6 study you mentioned here, Greg, great question. We're very excited about it. You know, you're the champ until someone knocks you out. And right now, in many respects, transplant is the champ in frontline therapy. But there's a pretty heavyweight battle going on with CAR T, and we may have that result in another year or so. Here's a great question from Jane saying, is it possible to get a recording of this? I'm in the UK and it's quite late for us. We don't expect everybody to stay up until 1 a.m. like Barbara. Yes, you will see in these slides that are also accessible to you that you can watch this video. If you go to the videos on the IMF website, this recording will be visible again. Here's a question. What is the difference between autologous bone marrow transplant and allogeneic bone marrow transplant? Is it true to perform allogeneic bone marrow transplant after failing more than one auto BMT or CAR T cell immunotherapy? How many times is it possible to do a transplant? Well, you're right. Auto transplant means you get stem cells from yourself. So we collect stem cells from you, then we give it back to you. Allo transplant is where you get stem cells from someone else. And that is not something we typically do as much as we used to do in multiple myeloma because it does carry a lot more risk and more challenges, and it hasn't been as helpful as we initially thought it might, although there's still some patients, especially in certain countries, where that may be the best option for them. But the allo transplant is still performed in certain places. We do think that much of that will be replaced by CAR T cell therapy. And then last question that I'll take for now, because we really have to wrap up, I'll hand it back to you, Robin, is what will be the impact of the REMS removal from CAR T? So you can learn more about this at the IMF website because it just came out and we reported on it, that there was a reduction in the requirements when people go through CAR T cell therapy. Things like the driving restriction that was at 8 weeks has now gone down dramatically to 2 weeks, that there are certain steps that the CAR T centers used to have to go through that they don't have to go through now when giving CAR T cell therapy in lymphoma and myeloma. And we hope that this will contribute to greater access to CAR T cell therapy. So many more questions, Robin. I'm sorry we can't get to them all, but I'll pass it back to you.
Well, thanks, Dr. Joe. And of course, Todd and Barbara and Michael, I think all of you together have made this an incredibly valuable and hopeful webinar in our world of myeloma. And so I just want to, in addition to what Dr. Joe has already let you know about, and of course this recording will be available on the myeloma.org website, there is another webinar coming up, and this one is especially focused on managing myeloma at relapse. So you can see the date and time here. You can see the wonderful lineup of myeloma specialists that will be presenting, as well as one of our nurse leadership board members, Rebecca. And so please mark your calendars for that, especially if you are in that relapse setting. And for these webinars online, they are very similar if you don't live near where one of our patient and family seminars are, or our regional community workshops are in person. Then that's why we have these myeloma community workshop webinars available online for you. And they cover the variety of topics from newly diagnosed all the way through relapse and clinical trial. So we hope you'll be able to tune into that as well. Here again is a reminder of where you can find those replays under videos. So please do check them out. And as Todd said, continue learning. The more we learn, the better questions we have for our healthcare team. And as always, we leave you with, we want to hear more from you. So once we wrap up today, don't just shut your computer down. Please wait for that pop-up survey to come in. But if it's 1 a.m. where you live, maybe you do want to go to bed, and no worries, get a good night's sleep. We will email you shortly after tonight's workshop. And however you do that, we appreciate you and all that you help us to make better for you. So we're concluding now with all of our gratitude to each and every one of you. But again, to this panel, to Dr. Joe for make it simple, right, Dr. Joe? So thanks to our sponsors, again, AbbVie, Johnson & Johnson, Regeneron, Sanofi, and thanks to all of you. We hope you have a wonderful evening, and just keep remembering our vision and our mission and the hope for all of our future. Good night, everyone.
Good night, everyone. Thank you.